cuando quieras.
So thanks everybody for this morning seminar. I think this is the first of the season, right? And just before starting, Emilia wants to announce
just to let you know that we have a new researcher with us, which is Gan Li. He's coming from Fuzhou in China. He's an assistant professor there. He will be working as a postdoc in some network problems, mainly with machine learning. What's it? Network. So he will be in the first lobby. Well,
so any other announcement? No. Okay, so it's a great pleasure to introduce you Michelle Thums from the Australian Institute of Marine Science. And the talk is part of a meeting that we are having together with Ana Sequeira, Mark Mikan, and Juan Fernandez, and also Carlos Duarte, with the idea of analyze megafauna, marine megafauna movement. And well, as Carlos is the director of the Oceans Institute there in Australia, in Perth, so I think it's more convenient if he introduces Michelle's background and explains a little bit what the kind of things they do there. And so, Carlos, please.
Thank you, Victor. So, thank you all for coming today to this seminar. And I'd like to introduce Michelle Thums, or Dr. Michelle Thums, from the Australian Institute of Marine Sciences. And Michelle completed her undergraduate degree in biology in the University of Sydney, and then moved on to the University of Tasmania in Hobart to conduct her PhD, which was on the movement ecology and the behavior of elephant seals, which will be, I guess, somewhat the subject of this talk as well. And those are very large animals that engage in very long and challenging migrations in the Southern Ocean. And upon completing that her thesis on this topic, then Michelle got a position as postdoc at the Udally Way Oceans Institute, but really on a partnership between the Australian Institute of Marine Sciences, the CSIRO, which is a bit like the equivalent of the SIC in Australia, and the University of Western Australia. We have a partnership called the Indian Ocean Marine Research Center, and Michelle joined that center as a joined the institute as a postdoc, and also Ana Sequeira, who's here, also is a postdoc at the IMROC. And within her first years as a postdoc in the IMROC, then Michelle developed a broad range of research topics on movement of whale sharks, which are the largest fish in a way in the ocean, and also the movement of turtles, with a particular interest on the small husks, the newly born turtles, and how they move into the ocean and navigate. And then also the movement of inanimated things like plastic debris in the ocean, and how plastic distributes in the ocean. And then last year, Michelle then got a position as researcher with the Australian Institute of Marine Sciences, but still engaged with the IMROC. And before I sort of read the title of her talk, which you can do yourselves, then just to explain that Michelle, Mark, and Ana are here in Mallorca on a meeting of a working group that we have established on understanding global patterns of marine movement. And there was, when I moved to Australia, there was an MOU signed between FESIC and the University of Western Australia to promote collaboration in 3 areas. One of them marine sciences, but one of them is computing-intensive sciences, big data, complex systems, like a package. And up to now, there have been no developments of the MOU in this area, and this project where Victor and Juan participate from IFISC is the first development in establishing a collaborative program between the University of Western Australia and IFISC on complex system analysis, in this case applied to understanding the movement of marine megafauna. So tomorrow there will be 2 more seminars in IMEDEA. About those 2 seminars will focus on whale sharks, and there will be a seminar by Ana Sequeira, I think starting at noon, on models to predict the distribution and presence of whale sharks. And then Mark Mikan will give also a seminar on a broad array of subjects of ecology, evolution, and maybe future prospects of whale sharks in the ocean. So with that sort of context of what we're doing here, then just I'll give the floor to Michelle to tell us about insights into the movement of marine megafauna. Over to Michelle.
Thank you very much, Carlos, and thank you all for your interest here today. So I'm going to talk to you a little bit about some of the insights into the movement of marine megafauna from my research, and also some of the insights from the field of movement ecology in general. So marine environments are very dynamic, and marine megafauna adjust their movement. They move long distances and they adjust their movement to acquire the resources that vary, often predictably in space and time. And their food resources and their breeding habitat can be separated by hundreds and thousands of kilometres, so this necessitates seasonal migrations often. The drivers of their movement might also be associated with optimal temperature and also optimal habitats. And these migrations may be repeated several times each year, for example in albatrosses, but also annually, such as the elephant seal does. It migrates from its breeding. These yellow dots are the islands where they breed and molt, and so annually they travel from the breeding grounds to Antarctica to their foraging grounds to build up their resources to fuel their breeding. So the early studies of animal movement relied upon just the seasonal presence and absence of animals appearing at a breeding site, for instance. And then later, people applied unique identification tags to the legs and flippers, etc. of animals. So here's a flipper tag on the rear flippers of the elephant seal. So you could work out when these animals turned up at the breeding colony, and they might turn up in other places. So that was how we learnt about animal movement in the earlier days. But of course, this couldn't tell us anything about their migration corridors, so what corridor they took to get there, and about their movement patterns. And it's really the advances in electronic tracking devices that have revolutionised our understanding of marine animal migration. So by far the most commonly used devices for tracking marine animals, or animals in general, global positioning systems and Argos satellite telemetry. And so here's some examples of some of these tags along the bottom. So these are the devices that we attach to animals, they're carried by the animal. And so as well as the satellite link devices, as are shown here, we also have these logging devices, and they can collect a whole load of really fine scale information on what the animal's doing. So maybe its heart rate, its stomach temperature, it might measure the water temperature, the 3-axis acceleration, the light, which we use later to work out where they've been. And then there's also acoustic tracking, which is depicted here. So you have these acoustic receivers that are placed in the environment, and then this method is commonly used on fish. So the fish have acoustic transmitters which emit a ping, a unique coded ping which is heard by the receivers in the environment. But this type of tracking is mostly used in coastal areas, but now we have networks of these receivers all over the world, so we can actually look at oceanic migrations using this method as well. And so now we have these really quite exciting new tags which have onboard data processing and compression capabilities, so they can actually transmit not just the location of the animal, but also the diving profile and ocean salinity and temperature data. And I've got an example of what this looks like here. So with your normal satellite tracking device, you'll be able to reconstruct the surface tracks of the animal. So this is in Antarctica now, on the Antarctic Peninsula. And so these tags are able to transmit information on the diving behaviour, so in the vertical dimension as well. And then they're also able to give us information on the temperature and salinity profiles. So not only do we know where these animals go, we know how deeply they're diving, and we know something about the environment in which they're diving in and foraging in. So we can really start to build up a really good picture about what drives the movements of these animals. So Argos satellite tracking systems have been available longer than any other satellite tracking technologies, and they are the most commonly used. So most of the data we have are from these devices. But for these devices to work, the antenna must be free of the water to communicate with the satellites. And so for animals that remain submerged, like our whale sharks, we use these tags, which are called Popup archival tags. So it's attached to the animal with this titanium dart here, and it logs all the information on board, and at a pre-programmed time, the tag, this is a link here, a weak link that will come apart, and then this is a buoyant device here. It will float to the surface and then transmit the data via satellites to us, the user, which we can then download later. So of course the tracking data are of immediate quantitative value to identify migratory pathways or home ranges, as can be seen here. So this is a sea turtle that's being tagged here at its breeding ground. This is northwestern Australia here, and it's just colour-coded by the number of days at large. So the animal's moved relatively quickly up to this area where it's then remained for most of its time. So that's really great. We can pretty quickly work out what's going on there, but there are many quantitative techniques to extract fine-scale information that were previously obscured by factors such as location error. And most of these analyses are based upon correlated random walks, but there are also some other ones, such as levy-flight methods. So by far the most commonly used quantitative techniques to analyse this sort of data are state-space models and hidden Markov models. And for both of these, they fit correlated random walk models that use the turn angles and the move lengths and the autocorrelation to infer the hidden behavioural states in the data. So we don't know what the animals are doing, just from the location data, and we want to be able to infer more of what they're doing from the characteristics that I've explained here. So the state-space model also handles the observation error in the animal tracking data, and that's to estimate the most likely location, and I'll show you how error-prone this data is in a minute. And it has two equations. So one of the models is actually a hidden Markov model. It models behaviour, and it accounts for the behavioural stochasticity. And the second model deals with this observation error. And so this is just the observation error that we have from Service Argos. So each location that we get, they tell us what class it is and the associated accuracy with each class. And you can see that it can get very bad. And in the early days, a lot of people just said, oh, well, we'll throw out these poorer quality ones. But for marine animals, we don't get a lot of data because they're often underneath the water and not transmitting any data to the satellite. And so we often get quite poor quality locations. So we need some better methods to be able to deal with this inherent error. And so this is just a slide to show you how this model works. So you can see the very high error in the raw data here. So this is Cape York in Australia. And this is the track from one turtle. This is its nesting ground at Rain Island, and it goes to its foraging ground. So very high error. So the position error is modelled with this observation equation, which assumes a t-distribution error with associated variance and degrees of freedom. And then we overlay the best location estimates, as we've done here. The colour indicates the inferred behavioural state from the autocorrelation to the previous displacement and the turn angle. So you can see the two modes here, red, which is nominally foraging, and so this has low values of autocorrelation and then high values of the turning rate. And then for the nominally transiting mode, we've got high levels of autocorrelation and low levels of turning rate. So using this sort of model, we can move beyond this sort of quantitative view that we had previously. So all we knew was where they went. So this is a plot of all the turtles that were tracked from this area. The different colours are just the different individuals that were tracked with all the error. So we can see where they went. That's nice. But now we've got some context. We know that this is the the breeding ground. So this is an area where they remain resident for a period of time. And then the black is where they've been transiting, and these other red areas are where they've been foraging. So then we can start to get an idea of the ecologically significant areas for these animals. So of course the breeding ground is a significant area, so this is now just a kernel smoother, and the warmer colours mean more points are there, so it's a more important area. But outside of the breeding ground, that of course we know is important, we now find that this foraging area here is very important, and these are some minor ones of importance. We can then start to look at the drivers of their movement as well. So not only understand these hidden states or what they're actually doing along their migration, but what drives their migration? Why do they travel it, and what drives them to go to these different areas? So here are some tracks now, again from northwestern Australia, of some turtles. There's 8 of them here, and they've been analysed with the Statespace model, so the red showing the foraging area and the black showing the transiting areas. And you can see straight away that they all follow a similar, some of the tracks aren't as long, but they all follow a similar migratory pathway, which is on about the 64 metre contour, I think. And then they all forage in a really similar area as well. And so we can then focus in on these foraging grounds to see, well, why are they going there? What's so good about this place? And we find that the turtles forage in this region a very complex topography of submerged carbonate pinnacles and banks and shoals. And so this sort of topography supports high diversity of marine species because it's got these suitable substrates for them to live on. And also the channeling of water around these banks creates locally enhanced productivity. So we now know that this is a very important area for turtles that we need to protect, and you can see that this polygon around here is actually a marine protected area. So we're obviously doing a relatively good job of protecting important turtle foraging grounds. Also what we noticed, well what we started to think about was the turtles followed the 64 metre depth contour, but if you're a turtle, how do you recognise depth? How do you follow a contour? So we started to wonder about that and why they were doing that, and we wondered if they were following the tidal fronts in the area. And so one of my collaborators is an oceanographer, and he was able to make a numerical model to determine where this tidal, or to determine the tidal current amplitude, and then using this equation we were able to determine where the fronts occurred. And so the black line is the tidal fronts and the red line is the turtle tracks now. So you can see it's not a perfect agreement, but it looks like they do seem to follow these tidal fronts. And when we looked at the chlorophyll map for the area when they were travelling along here, we can see that there's a pretty good match with what's happening with the chlorophyll. So it looks like they did focus their transit in the vicinity of the tidal fronts and also the foraging area as well. So what would this be? So the tidal front's thought to be a region of convergence to enhance the foraging habitat for these animals, and they use the fronts maybe to aid as navigation to the foraging grounds in an otherwise homogeneous ocean. So maybe that was the reason. Of course it's pure speculation at this point. We're going to do some more modelling to determine whether this is the case. So now I want to switch tracks and focus a little bit on the vertical movement behaviour. So I mentioned before that the tracks can collect, the tags can collect information on the diving behaviour too. So here are some tags on these very fat pregnant elephant seals at Macquarie Island. And these are the sort of tags that we need to recover to download this data. And because the animals come back at predictable times and to the same location, they have very strong fidelity to their breeding grounds, we can actually recover 80% of these tracking devices. And so this is just typical depth recorded by a tag for 1 week, depth on the y-axis there. And you can see the animals spend very little time at the surface. This is an air-breathing animal, it's a mammal. But it just dives and dives and dives, and it does that for its whole 8-month trip. Its average surface interval is about 2 minutes. And so you might wonder when these animals might rest, and it's thought that they rest in the water column by doing these drifting dives. And I've shown some of them here, in between some of these dives that are thought to be foraging, where they go to a depth and wiggle up and down and come back. And we see these every day of the record, and sometimes twice a day in a sequence like this. So they're dives where they swim to a depth, then they drift passively in the water column, and then they ascend very quickly again to the surface. It has a very characteristic time depth profile, which I'll show you here. So that's the ascent, the drifting phase, and then the descent, the drifting phase, and the ascent. And we know that they're not swimming because we also measure the speed of the animal. So you can see here the speed is 0 during this drifting phase. And of course the animal isn't just completely stationary in the water column, but we measure speed with a turbine, and the animal's swimming so slowly that the turbine stalls, so it measures 0. So we've got this active swimming component and then the passive drifting component as well. And that was just to show you what this tag looks like. They're the turbines that measure the speed. So why are we interested in these things? So fluctuations in the body composition, so the ratio of the fat to the lean tissue at sea can lead to measurable fluctuations in their buoyancy. And the buoyancy, and hence the drift rate, will depend on these ratios of fat to lean tissue. So the fatter the seal, the slower the drift rate. So we can tell something about changing drift rate about the body condition of the seal. But so in order to validate this, we need to calculate the percentage body fat of the seals. And so here are just a few pictures of how we would catch them. And this is a tripod and with a block and tackle. So we would hoist the animal up to weigh it, so we could get the mass of these animals. They're about 600 kilos on average. So first we sedate them and we roll them into the net. Then we hoist them up, measure them, measure their length, and then we have an ultrasound here where we can measure the thickness of the blubber. And so from all these measurements, we were able to calculate a percentage body fat. And then we looked at the relationship of the body fat with the drift rate, the drifting rate of the seal. And you can see a very good relationship with an R-squared of 0.9. So the drifting rate depends on how much body fat these animals have. So if you're a fat animal, you'll float, and if you're a skinny animal, you're going to sink. So we were able to use this as a measure of foraging success of these animals, because we really can't tell anything about where they're successful. We only know where they go. We don't know where the productive areas of the ocean are. And we can only gather that the areas that they go to are productive, but of course they may not be. And there may be differences in the productivity of the regions they go to. So what we were able to do then is predict the fat content throughout the whole foraging trip. So here is the predicted body fat on the y-axis, on the left-hand side, and on the right-hand side the drift rate. We're able to fit a spline model to the pattern of change in fat over the whole trip. And then by looking at the change, whether it was positive or negative, we could see whether they were gaining weight or whether they were losing weight. And so then, of course, we could look at where, spatially, in the ocean, they're gaining and losing weight, to look at these most productive areas. So if you just focus up here now, I've just used one of these kernel smoothers again on the points, so the warmer colours are where there are more points. So this is a map of where they've been in this searching or foraging mode and where they've been gaining condition. This is Macquarie Island, where they were tagged. And so you can see not much happened in between in terms of gaining mass, all of it on the coast of Antarctica and in the Ross Sea. If we focus down here now, where they were in their transiting mode and when they were losing condition, you can see that this whole area, which encompasses these major frontal zones of the Southern Ocean, are not very productive for these animals. They swim through there very quickly and they lose condition. So now a little bit about some other animals' diving behaviour to look at how we can work out what the drivers of their movement are. This photo is very dark, but this is how we get these tags onto the whale sharks. These animals are very large. We work with, they're about 10 metres in length. They can get up to 15 metres, so as Carlos said, the biggest fish in the sea. And so we use these tags. It's got this tether that's attached to a titanium dart, and using a pole spear, we can attach it to the animals. And so if we recover these tags, which we do very rarely, then we can get this very nice diving data. So again, you can see the animal diving to depth and coming back to the surface, and the grey bars are just nighttime. But you might wonder why a fish needs to come back to the surface. It doesn't need to breathe air. And so we wondered why the sharks do this. All day long and all night long, they go to depth and come back to the surface. And so the hypothesis is that when the sharks go to depth to feed, they get cold because they're exothermic. Their body temperature is the same as the external environment. So they cool down, they have to come to the surface where they might warm up and where there might be more oxygen as well. So we looked at whether there was a relationship between the time that they spend at the surface and the minimum temperature that they experience on the bottom of the dive, because these tags also collect information on the water depth, the water temperature. And so what we did find was that there was a relationship. So the more time, so this is the medium surface duration. So when you have a high surface duration, you have a very low temperature that you've experienced at the bottom. And of course, when the water temperature is very warm, this relationship breaks down a little bit. So it mostly is happening in deeper water when the animals are in the pelagic zone. So now I just want to talk about some future direction. We have, I've shown you a lot of tracking of just individuals of separate species, not very high numbers. And the reason that's the case is because these devices are extremely expensive. They can be in the order of $7,000 each. And then the satellite time is also very expensive. So we typically work with low numbers. But now, because our research area, or the researchers in our area have been using these tags for a very long time now, there's starting to be quite a lot of data, and this graph just shows the number of deployments of tracking devices on the y-axis through time from 2004 to the present. And these are just the different types of animals. In the dark blue is marine animals, in the green is terrestrial animals, the orange is birds, and this is the total. So you can see 720,000 tags have been deployed, and we also have data previous to this. There's been satellite tracking done on animals since 1998. So we're now really moving into the area where we have big data, just like you guys do here. I know a lot of you are analysing human movement from the tracking that can be done using smartphones. And so we're hoping now that we're getting into this order of data that's needed to be able to now use some complex systems approaches to be able to understand what these animals are doing. So this has already been done in the North Pacific. So there's been a big collaborative effort where all the researchers have pooled their tracking devices, so 4,300 in total on 23 different species. Lots of tracking days over about 9 years, and these are all the different guilds of animals. You've got fish, you've got seals, you've got sharks, you've got birds, turtles and whales. And so this is what all their just raw data look, or not raw data, but all their locations look like and where they go. So you can see we're covering the whole area of the ocean now. And if we did this for the whole world, we would cover every ocean. So really that's what the future direction is, and that's why we're here now working with Victor and Carlos and Juan. So now that these tracking data sets have achieved big data status, there is potential, but they're not publicly available like much of the human mobility data is. So So that's one of the things we're also working on, is trying to collaborate with a range of different ecologists to try and bring this data together and make it publicly available. So we really, as I mentioned, we want to try and use a complex systems approach, as has been used on human mobility, so that we can examine the interactions between species, because we've never been able to do this before, and it's only with these very large amounts of data that we will be able to do this, and that we can perhaps describe these universal laws of mobility, as has been done for humans, and identify species based on their activity patterns, and then look at the temporal and multi-layer networks as well. So I think there's a lot of opportunity here if we can get access to these big data. So I'd just like to leave you with this very pretty photo of a whale shark, and to thank Carlos Duarte and Victor Aguilas for hosting us here. Thank you.
So thanks, Michelle. So there is plenty of time for questions.
You have shown that some cases the sea lions go down without swimming, just basically. I've read that in some cases, so I don't know if they always sink, so they cannot, they should swim to remain at the surface, because I've seen some, I remember something I've read that sometimes they also go freely but in the horizontal. This implies that they don't go down. So I don't know, if they don't move, do they fall down?
So most of the time they are negatively buoyant. So when they stop actively swimming, then they will sink. But sometimes, and well, it often happens towards the end of their migration, they become positively buoyant because they're very fat now, and then they will rise up when they stop actively swimming. Yeah. Yes. So we also see that as well.
This is because there is some correlation between the motion, the migratory motions, and the currents in the sea. But this depends, of course, if they swim in the vertical.
Yeah. So they swim for the first part to get to some depth, maybe around 150 metres, and then they stop active swimming then, and that's when they're resting. So they're sleeping while they're drifting, and so people put accelerometers on the elephant seals as well, and they found during the drifting phase that they were actually spinning like a falling leaf, down, 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 until they get to some depth, and then they actively swim again to the surface.
It was on the turtle case, you showed some chlorophyll maps.
Chlorophyll maps?
Yeah, and then I'm not sure, but are the turtles plankton? Are they?
No, they don't eat plankton.
And the next question is also the front. You show the front.
Yeah.
What could be the advantage of a turtle to swim within the front or outside the front? What's the hypothesis behind it?
Yeah.
For the chlorophyll, for the front?
Yeah, so the hypothesis, there could be two explanations. So one is that because it's a convergence zone between the mixed water and the water that's not mixed, so it could be a zone that is the turtle might perceive, and it might be a signpost for them to follow to their foraging grounds northward. And also it is an area of enhanced productivity where these two different water bodies meet. So they might also be foraging en route on things like jellyfish. They mostly eat benthic invertebrates, but they also eat some jellyfish. So it might be an area where there's some enhanced productivity as well.
So they do eat in the water column as well? They not necessarily eat on the seabed.
Mostly on the benthos, but also in the water column.
More questions? So, well, I understand that just storing a lot of data about positions and all that can be very helpful in order to have a statistical description of places with larger residency or these kind of things. But I think that in one of your transparency you saw basically something that we don't typically do, is is trying to have an ecological understanding of why.
Yes.
Right? And for that you need other kind of maps, like topography, salinity, chlorophyll, in order to have an explanation of that.
Mm-hmm.
And this is not much done when you do it with human mobility. So my question is, as a biologist, how much value has a statistical description for you, or you really need to have this other ecological explanation of the findings? And
Yeah.
then this kind of data is also valuable at the same scale.
Yeah.
That will be the next.
Well, I mean, for us, we need to understand not just where they go, but why they go there. So in order to understand that, then we need to have these environmental data. And it's also very important for predicting responses to climate change. If we know what drives their movement, then we'll be able to predict what might happen under some different scenarios.
Because the complex system approach will tell you, okay, you have a place where it's highly visited, it's a hub or things like that, then if
Yes.
this place is affected, then that will affect the whole ecosystem, right? But it's not introduced one. Sometimes you do, but mostly you just rely on the statistical description. So I wonder whether this is satisfactory for you, or if you are asking for more.
Well, we would always do both. We would always want to have some statistical way to analyse the movement patterns, but also to then overlay whatever those patterns are with whatever environmental data we can get. Does that answer your question?
Yeah.
I mean, it's somehow related to that also. At the end, you said we're looking at deriving universal law of movement of animals. Then you were showing, I mean, there was birds and large fish and mammals, and I'm guessing they don't have the same life cycle, they don't look for the same food, they don't necessarily have the same ecological range where they live and things like that. So, I mean, how much can be universal and how much will be species specific?
Yeah, well, I mean, they all have the basic requirements that they move from breeding grounds to foraging grounds.
Right.
And many of them have similar thermal regimes, thermal environments that they find more preferable. So they're driven by the same sorts of things. So we would expect often, not all the time, of course there are animals that do very long migrations and there are animals that do very short migrations. So we wouldn't necessarily expect always that a law might be universal for the different types of movements that we see. We think it would be interesting to look to see whether we can find some universal law to describe their movement.
I mean, I'm being a bit provocative, but in these maps of the turtle, where you were showing the foraging ground and things like that, maybe you just look at nutrients or chlorophyll and then you realize this place is rich and you don't necessarily need to have all the tracking thing. You know what I mean? If they would have some universal law, then maybe you just look at the physics and the production there.
Yeah.
That is telling you which place are important.
But if you saw on the chlorophyll map, they didn't go to the areas where there were the most chlorophyll.
Right.
They went to the transition zone between the areas where it was profitable and where there was more productivity and where there was less. So it's not always so simple. And always, as you pointed out, I think earlier, these animals don't eat chlorophyll. Right. Well, they don't eat, zooplankton or things that might be closely related to chlorophyll distributions. So often, even though chlorophyll can show us areas with higher productivity and we might expect other animals like fish might be present for other animals to eat them, often there are time lags in this information, and the spatial resolution isn't measured, it's not measured on the same spatial resolution as the animal tracking is. So we often don't find relationships, even though you might expect them.
I mean, there is this, this sort of hotspot vision in the community, saying when there is the physics, there is the plankton, and then there is everything there,
Yes.
So, I mean, I don't know, maybe it's true, maybe it's not true, I'm not sure.
Yeah, It can be, but like, yeah.
Between the
Yes.
different level of the trophic chain.
Yeah, there are relationships, but they're often weak because of this spatial resolution mismatch
Yeah.
between the data. Yeah.
Can I also say something?
Sure.
I think part of the things that we are interested as well is in describing the movement. So even if those chlorophyll patches would be the ones that they go to, we want to understand how we find them as well. So we are interested in that sort of.
Yeah, not only locating them, but also how they move to
Yeah, how do they find them?
Yeah.
So there are somewhere here they do a 1,000-kilometer migration.
Right.
How did they know that there would be food in the population? Is that correct?
Yes. Yeah.
But the same argument you can do it for humans, right? You know where the houses are, you know where the people live, and you know where the offices are and the industry, you know where they go to work, right? But this is not the whole story, right?
Yes.
There's a map more. And there is another effect, there is a feedback between the two things, right? In humans it's clear. So you go to the city, more people go to the city, more opportunities, and then you need to develop them. But also I suppose that in hotspot, right? You have a place where a lot of animals go for feeding, that would deplete that area, right? So so that could be also an interaction between them. So that's the two things.
might change.
Yeah, and I think that the abundance of resources is, I think that's the important ingredient characterizing the mobility. Because if you don't have problems to feed yourself, you basically don't care. But if the resources are scarce, this is asking a lot for you, right? To develop strategies and all that. So I think that's probably more, fitness of.
Also, they have a specific searching behavior, and there might be some sort of influence from human impacts, for example, shipping routes or something like that, interfere with that. So if that is broken, then they might then find that resource.
Right,
So we need to try and understand it.
Something else?
Please.
And maybe it's important also to specify the scale and the focus, because, for example, the works you have shown, some part is more to the behavior biologists' interest, like the movement pattern and the way an individual moves.
Mm-hmm.
But then we talk now about chlorophyll, and this is more to ecology.
Mm-hmm.
So, and also the scales, temporal and spatial are different. And so there it's not a sole question. I think it's a range of disciplines and scales that you can search. It's a diverse topic. I mean, a behavioral biologist maybe will be interested in the movement pattern, if it is correlated or not, about the turning angles, but the ecologist maybe will not care about this, will care about the long range.
No, they do. I am an ecologist. Because we want to know where they're spending the most time. So, I mean, it's just an objective way to do that, to understand, to be able to objectively determine the transiting or the migrating phase from the foraging phase, because there will be different threats in each of those places as well. Like Anna mentioned, if shipping zones are in the area where they have these migratory pathways, and for the turtles I showed you, it was the same one. They all followed the same migratory pathway. But for the elephant seals, it was more dispersed. So it is very important for us to be able to objectively delineate between those two types of movement patterns, and so that then we can focus on where the ecologically very significant and important areas are. And then we could also look at the underlying environmental data in those regions to see what drives their movement when they're transiting, and what is the important area for their foraging ground. So for conservation, it's very important to be able to protect their foraging grounds, of course, because if they're not having successful foraging, they won't be returning to breed, and they won't be successfully producing offspring, and their populations will decline.
Okay, so time for our last question, if any? No? So, thanks, Michelle, for